Welcome to today's video. We'll be looking on molecular medicine and its basis. A gene is a functional unit of genetic material and is located on the chromosome. Genome is a complete set of genes. Genetic information are stored in nucleic acids. There are two major types of nucleic acid: DNA, also called deoxyribonucleic acid, and RNA, also called ribonucleic acid. Now let's look on the functions of these molecules. So DNA functions as a storage molecule as well as transmission of genetic material, while RNA has broader functions. There are many types of RNA: ribosomal RNA, rRNA, messenger RNA, mRNA, and also transfer RNA called tRNA. Now let's look on the difference between purine and pyrimidine. So nitrogenous bases are present in nucleotides. These are adenine, guanine, cytosine, thymine, and uracil. Purines have two rings. These are adenine and guanine. Pyrimidine has one ring, and these are thymine, cytosine, and uracil. DNA is different from RNA in that it has thymine while RNA has uracil. Now let's look on the difference between a nucleotide and a nucleoside. A nucleotide consists of a nitrogen base, a pentose sugar, and a phosphate group. A nucleoside has a nitrogen base and a pentose sugar. There is no phosphate group. Now that we know the difference between a nucleotide and a nucleoside, let's look on some examples. So the nucleotide or the nucleoside, they are named based on the nitrogen bases. Example, adenine, the nucleotide is adenosine and the nucleoside is adenylate. For guanine, the nucleotide is guanosine while the nucleoside is guanylate. For cytosine, the nucleotide is cytidine and the nucleoside is citidylate. For uracil is uridine and uridylate, and thymidine and thymidylate. Now let's look on the DNA structure. So the DNA is a double helix. It has 10 base pairs per helix and two grooves, the major groove and the minor groove. Its bases are separated by 3.4 amperes, and the diameter of the helix is 20 amperes. Within the DNA structure, there are many bonds. 5' phosphate group on a nucleotide joins to the 3' OH group on another nucleotide. Also, the OH group on the sugar, which is within the nucleotide, binds to water to form hydrogen bonds. At pH 7, there is a negative charge on the phosphate group. The positive proteins that exist neutralizes these charges. There is also a sugar phosphate backbone that is on the outside of the molecule, while the bases are on the inside. The strands are anti-parallel and there is complementary base pairing. That means A binds to T and C binds to G. When A binds to T, there is a formation of a double bond, but when G binds to C, there is a formation of a triple bond. In molecular medicine, there is an important rule that is called the Chargraff Rule. This states that the percentage adenine is equal to the percentage thymine or uracil for RNA. The percentage guanine is also equal to the percentage cytosine. That means that the percentage purine is equal to the percentage pyrimidine. Let's look on an application. So if a strand of DNA has 35% G, what percentage T is present? So if the percentage G is equal to 35, C is also 35 according to Chargraff Rule. This makes 70, and therefore 30% of adenine and thymine exists. If we divide this by 2, we get the percentage thymine. Now finally, let's look on DNA packaging. So when DNA is packaged, it binds to histone proteins. DNA plus histone proteins are called chromatins. There are 2 copies of each histone protein, H2A, H2B, H3, and H4. These form an octamer. The octamer of histone proteins, along with the DNA, are called nucleosomes. There is another form of histone that is called the H1 histone protein or linker DNA. It joins the nucleosomes